Okay, so I will talk about exploring personalized cancer treatment by integrating digital twins and QSP modeling. So the outline of the talk would be, I will talk about our team and then objectives and some of the models that we created, and then the next phase and some of the challenges we are facing. So this work has been initiated by NCI DOE Ideas Labs on digital twins. So I participated in July 2020, a meeting that was basically 5-day virtual Ideas Lab toward building a cancer patient's digital twin. It was sponsored by both NCI and Department of Energy. And the event brought together a diverse group of researchers from computational side, as well as biologists and physicians. And in late 2020, 5 of the team projects were selected for the funding, and then 3 more were funded additionally through Department of Energy. So our team, we created a proposal for this, and it was funded by both NCI and then Department of Energy. So it was one of the 3 teams that was elected to get the funding from Department of Energy as well. So our team had a diverse people from computational background, and me and then Naveed. He was my former postdoc. He's currently at University of Columbia. Pamela Jackson at Mayo Clinic, Venray Howe at Penn State, Ali Reza Sadpour at UMass Dartmouth at the time, and Yang Huan Chang at University of Oregon Health and Science. And from biology and experimental sides, we had Yanis from University of Pittsburgh, Adrian Lee from University of Pittsburgh, and Colin and Mohammed and Daniel from Ohio State University, and Zuzana at the time, she was at OHSU, and Eva Katsulouskis from Veteran Hospital in Tampa. So we came up with this idea of creating such a digital twins that have a database that include both including DT's output and the other data sets. That include biomedical data set that are collected through the time, and then biological data that are available and it have been collected through the time, and EHR data of cancer patients. The computational core includes mechanistics modeling, machine learning, and some stochastic simulations. And the main computational part, the idea is using QSP modeling each organ separately and modeling the connections between organs through both mechanistics modeling and machine translation models. And this is a dynamic, always they fit to each other, each of these models. And then the output of the DT would be data collection guideline that tell us what type of the data and when should be collected for the patient. So it says that, okay, until this time, I have a good prediction of the patients going on. After that, we have the higher uncertainty. So you need to do surveillance and do the collections of the data, what type, blood work or whatever is just needed, or images, what type of the data and when should be collected, predicting the cancer evolutions, and then also interactive visualizations of how different organs and the tumor are evolving and the cancer in general is evolving. And then some validations comparing the data to what is observed after collecting the data. If it's not acceptable, the DT should tell us that what type of other data should be collected or how we should do this improvement of the model and the computational core and the data that might be needed to be collected to improve the prediction. Any question here? Okay, so as I mentioned, the goal was creating a QSP model that's modeling each organ separately, and then connections between the organs are modeled based on machine learning and mechanistics modeling. So at first, we start with the tumor. So we start with modeling the tumor at the pilot phase that we started. That's basically, we started with breast tumor because most of the people in the group had the specialty in breast cancer. So we looked at the gene expression data. Our team had developed before a TumorDecon software that was supported by NCI ITCR program. And then we get the initial conditions based on the TumorDecon, and then we just write chemical interactions formulas, and then look at the evolution of the tumor. In the next phase, we can do more data-driven mechanistics modeling by collecting MRI images and pathology images to look at the exact location of the cells in the tumor and the exact boundary of the tumor to model them, and then modeling based on the chemical and physical interactions between the organ, the tissue of the organ, and the tumor, and then looking at the evolution of the cancer. And then the next phase would be modeling the metastatic organ and their connection using both mechanistics modeling and as well as the machine translation. And then completing the DT with its database and provide it as a software by modeling other organs after that, and so on. So that was our vision. That's, I believe it's, you could say like a sci-fi vision, but it's something that I think with collaboration with everyone and the teams like you guys, it's possible. It's not something impossible to do, but yeah. So we started the work for the pilot phase. The pilot phase, we started with very small network. If you say to biologists, for the biologists, very small network, because the tumor has many interactions, many different cell types, and also cytokines. But we only pick those that are important because mathematically, this is really large network, considering that many unknown parameters and many parameters that we have to basically estimate. And also doing down synthetic quantification, sensitivity analysis for such a large system, mathematically, computationally, it's expensive. But for the biology side, we are just looking at a very small network. So we start with this network. If we needed, if the basically the prediction doesn't match with the observation, and so we can always add or change the model, the network, and add more complexity if it's needed. But to avoid too much complexity, we just start with this one. We model the interaction between cells and molecules using the basically mass action law. So we wrote a system of equation for changing each of the variables in the models, like that cytotoxic T cells, C helper cells, and dendritic cells, and so on. So all of these are the variables of the models, and IL-6, IL-10, IFN gamma, and so on, estrogen and HMGB1. These are the cytokines and the molecules that we have modeled in this. We use the digital cytometry methods. What is the digital cytometry? Because estimating the different cell types in the tumor is very expensive and time-consuming, and it's not a common data that has been collected because of those two main reasons. Methods such as these digital cytometry methods have been developed through the time. Decon RNA-seq was one of the initial methods, and then CyberSort and two other score-based models. These are the examples of this model. There are some other methods as well, but these were the methods that our team studied for our NCI ITCR project. And we developed a TumorDecon package. TumorDecon, it's basically a Python package that includes all these 4 methods. We also compare these methods, and we found that the CyberSort and CyberSort X B mode are the best options. But so the TumorDecon, which is a Python software, it gets the input gene expression data. And for some of these methods, like CyberSort and Decon RNA-seq, you need the gene signature metrics. So this one, we have also a method to create a gene signature metrics, or you can basically upload or select one of the gene signature metrics that we have on TumorDecon. We do pre-processing by basically matching the gene expression data, because as you know, some data are based on the Hugo symbols, some data are based on the gene IDs. So we will do these matches. The software do the pre-processing and some batch correction if needed. So you can say that if you need to do batch correction or not, the software will do. And then you select your digital cytometry method, and then it give you some post-processing result. That's basically from the gene expression profiles of the tumor, you get the percentage of different cell types in the tumor. So what the digital cytometry does is that get this gene expression of mixture data that includes many different cell types. And from the gene expression data, it gives you the percentage of different cell types in the mixture data. And then there are some visualization results. So if you like the TumorDecon, we have very nice tutorial on my website. If you go to the software pages and select TumorDecon, you can see the documentations, and then you can basically follow up the documentation to use the software. We looked at the, we applied this cybersort data, cybersort method on TCGA data and mass cytometry data. And we saw that, we applied on TCGA data and we compared with the mass cytometry analysis of the large cohort of the patients that have been done in another study. And as you can see, the results are very similar. So the most abundant cells in the tumor was macrophages, and then CD8 plus T cells, and then CD4 plus T cells, and so on. So the results were very similar to that. And also, so we did, going back to our breast cancer project, we used TCGA and metabaric data. We applied digital cytometry methods, CyberSort XP mode specifically, and we found that we clustered the tumor based on their percentage of the different immune cell types in the tumor. And we found that there are 5 distinct clusters of the tumors based on their immune profile. So for example, cluster 1 has high M0 macrophages, while cluster 5 has high mast cell resting, or cluster 3, as you can see, has high percentage of M2 macrophages. So we divided the tumors in these 5 clusters, and then we developed the ODE model for each of these clusters. One of the main challenges, lack of time course data for cancer patients, right? If we want to find the parameters of the model or validation of the model, always the main challenge is lack of time course data. So as a result, parameter estimation is a challenge. So we found some of the parameters by doing literature review. What we made also, we made some assumptions, like assuming the large tumors are at the steady state, they cannot evolve more because there is no space to evolve. And we made some assumption about the relationship among some of the parameters based on some biological studies. And then we performed some sensitivity analysis to find the most sensitive parameters, and based on that, try to find better estimation for the most sensitive parameters and do they again. So we found that for cluster 1, the most sensitive parameters, for most of the clusters, the most sensitive parameters were related to the cancer. So killing rate of the cancer cell by cytotoxic T cells or IFN gamma, or the proliferation rate of the cancer cells were among the most sensitive parameters. But after that, we found that if you set aside the same parameters related to the cancer, then we found that the most sensitive parameters were related to adipocytes. So this was coming very interesting, because some of the other studies that are medical studies and biomedical studies, they show that adipocyte has a really important role in controlling cancer, breast tumors specifically. So we found that also adipocyte, the parameters related to adipocyte are the most sensitive parameters for all clusters. And we varied we found that 2 of them, our assumptions are very important. And then we basically looked at these assumptions, we varied them and see that how the dynamic changes over the time. And for example, if we target IFN gamma, the death rate of the cancer cell by IFN gamma, you can see that it can have a really huge effect in one of the cluster, that's cluster 5, but it doesn't have much effect on cluster 3, the patients in cluster. So what is very important in this conclusion here shows that, for example, another one here, as you can see in cluster 5 has effect, but not in other clusters and so on. So what it shows that basically targeting tumors or the treatments might completely depend on immune compositions of the tumors. So in one tumor, targeting one of these can have a huge effect, but in another tumor, cluster of the tumors, targeting that specific parameters that would basically right now I'm talking about IFN gamma, the kill rate by IFN gamma, you can see that it doesn't have much effect in some other clusters. So we also looked at the mice tumors because, as you know, many studies do the experiments on mice and they try to see basically if they can validate the same observation in human after it's come to the drug development and so on. So the main studies at first, it's mostly done on mice. So we found the time course data of the mice breast tumors. And basically, we looked at the gene, it was the gene expression profile. We used the CyberSort XP mode to find the percentage of the different cell types. And then we used the curve fitting basically to fit the data of the evolutions of the cancer and other cell types in the tumor. And then we found the most sensitive parameters for this one. And we found that interesting for mouse, after the parameters related to the cancer and some adipocytes parameters coming up, but the parameters related to IL-6 come as very important parameters rather than mostly coming from adipocytes. So IL-6 parameters. And if you look at the studies that suggest targeting IL-6, it's coming most from mice experiments studies, but not much in human has been validated. It doesn't have that much effect in human tumors. As you can see that we see these disparities in the result as well. So for human, we see mostly adipocyte as the most important, but in the mice tumor, we can see IL-6 comes up as one of the most important parameters after the cancer, after the parameters related to cancer. Our collaborator at OHSU, they basically collected very nice, valuable data that's basically looking at location of different cell types in the tumor, from early stage tumor versus to the latest stage tumors in mice experiments. And then because for the simplicity, we only modeled this circle that you can see, the largest oval that we could fit and include all the cell types. And looked at the evolution of the tumor over the time. And our colleague at OHSU, Yang Huanchang, he basically used the image data and gave us the location in the grid, and also he analyzed the co-locations of the different immune cells, the distance between immune cells and cancer cells. So we converted our ODE model to the PDE models to considering the location and adding the locations and co-locations, modeling the spatial interaction of immune cells and cancer cells. So we had the advection terms that we had it and the diffusion rate of the cancer cells. We used the free boundary to let the tumor grow over the time. So we used Robin free boundary condition. And we looked at how the tumor evolves over the time. As you have seen in the experimental data, you can see that in the early stages, there are some immune cells, as you can see in the data, but when it goes to the later stages, the immune cells are depleted over the time, and it's happening to adipocyte. Adipocyte, we have some small number, but if you look at the other immune cells, it's basically gone, and then mostly it's the cancer cell. And what was interesting was that we observed the same thing. But very, very important to note that here, we didn't do curve fitting to this data. We did the curve fitting for the data that was collected from another study only for the ODE model, for this study. That was basically the mouse model that they collected the gene expression profiles of the tumor. So we didn't use this data to fit the models of the parameters of the model to find the parameters of the model. But interestingly, the observation of the model matches the observation of the experiments. And so we looked at the influx of immune cells and without influx and with influx of the immune cells. And we saw that if there are no flux, that was in the case that there was no treatment. And in the scenario of our study, the experimental data, basically immune cells are depleted. But if there is a flux of immune cells, then you can have some immune cells in the tumor after the time. And then we did the sensitivity analysis, and we found that the most sensitive parameters related again to the diffusion rate of the cancer cells. These are the sensitive parameters related to the PD model. Oh, there is a question?
Yes, I actually have a question regarding the advection model you use. Basically, for radiopharmaceutical strategy, we found that advection term is actually negligible. I was wondering if you came to certain conclusion in your research as well. And
So
also my second, yeah,
go ahead.
and my second question is that how you came up with selecting boundary conditions? Is there any specific reason to select this one?
So advection term, we assume it's negligible for cytokinase because the movement of them because there are small molecules. If you assume that you are looking at pharmaceutical terms, if you are looking at the molecules, we can assume that they are negligible because they are small particles moving around comparing to cells that are moving around. So the advection term we included for the cells, but not for the molecules. That was really good to mention that, and thanks for it. The other thing was coming from the boundary condition. We wanted to something that's basically let the tumor grow over the time. If you mean the special boundary condition that we used the Robin boundary condition, because we wanted the tumor would be able to grow or basically shrink if they do, but they didn't. So mostly it's growth. So that was the reason.
Okay, thank you. Please continue.
Okay, so we looked at the sensitive parameters, and then we found that the fusion rate of the cancer cells, and after that, the adipocyte is the most sensitive parameters. And then for the influx rate, we found that the influx rate of the cytotoxic T cells and then macrophages is very important. And then the influx source, that how much there are outside of the tumor that they could income. So cytotoxic T cells and macrophages become at first cytotoxics and then macrophages. And then sensitivity to reaction parameters, the same as before. Cancer cells are very important, the death rate and proliferation rate of the cancer cells. So basically, going back to the data-driven QSP modeling idea is that we have the primary tumor. We get the gene expression profile. The reason we use gene expression profile, as I mentioned, because it's one of the most common data that has been collected. There is a TCGA data. There are many cancer patient data that we have that they have gene expression data, but we don't have the exact same, the number of cells in the tumor, the composition of them. So we use the gene expression data, we give it to the digital cytometry method to estimate the percentage of different cell types. We cluster the tumors based on their immune profiles. And after that, we find the interaction between cells and molecules in the tumor, the key interactions. And after that, we built the mathematical model based on the interaction. And for each of these clusters, we find the parameters of the model. And the next step would be adding the treatment to the model. So after that, for each cluster, we add the treatment and we look at how the treatment would change the dynamics of the tumor in each of these clusters and try to find the optimal dosage for each of the clusters. So we did similar thing for colon cancer, and we looked at, we found based on the literature review, the key interactions, and we drove a system of equations for these key players. And by reviewing many studies, we just, as I mentioned, wrote key players. And as I mentioned before, we can add complexity if it's needed after that. And for each of the clusters, so we cluster patients based on their data. And for each cluster, we find the parameters of the model. So this is the colon cancer data. So it should be around 500 patients. So we looked at their immune profile. These outputs are also from TumorDecon. If you like the TumorDecon, you can basically, these visualizations, you can find it, use the TumorDecon software to create these. So we looked at the patients' gene percentage of different cell types in the tumor. We cluster them based on their immune decomposition. And then we developed the ODE model, as I before, challenge, lack of time course data, and then similar to the ODE model that we did for breast cancer, similarly, we find the parameters and perform sensitivity analysis on non-dimensionalized system of ODEs. So we found the most sensitive parameters. Again, it was mostly related to cancer. And then if you set aside the cancer parameters related to the cancer, we found that for most cluster, macrophages are very important. But interestingly, in one of the cluster 4, T helper cells and Treg cells, specifically the parameters related to Treg cells, become very important. And then cluster 5, again, macrophages and T helper cells become one of the important sensitive parameters. And we look at the dynamics of the patients in dynamics of the cells and molecules for each of these clusters. And then next step, we also looked at the spatial modeling. So this data that I'm showing in the right side is the data that was collected from another study, but the exact cell locations. And then we used our own model that was ODE based, and the parameters was derived based on the gene expression profiles. And we tried to see if we can see the how location of different cell types changes the tumor growth. Basically, assuming if the cancer cells are here, macrophages are close to cancer cells, how it would evolve if the macrophages are different locations, or for example, cytotoxic T cells in another location, the co-location of these cells, how they interact, and how would the tumor would be evolved over time. So I will show you a movie here. I hope we can see. So what you can see here, that what we observed here, it was co-localizations of macrophages and cancer cells among many different simulations we have done. We observed that it seems that cancer cells and macrophages, they feed each other, they help each other to grow and expand over time. So, and they become more close to each other in the places. And then we looked at the sensitive parameters related to this spatial organization of the tumor. Basically, look at first the sensitive parameters to the cancer. We also looked at the cancer, the parameters that are sensitive to cancer to immune cells ratio, and macrophages. Those that are in red box, there are the parameters that are in intersection between all these 4 subplots. We also looked at the parameters related to the velocity of the changes, the velocity of the tumor that would evolve over the time, so sensitivity to the velocity vector, and then also sensitivity to the W, that was the angle of the changes of the position of the mesh, basically. So, and as you can see, the diffusion rate of the macrophages is one of the most sensitive parameters here. So diffusion of the immune cells mostly coming like diffusion T helper cell and Treg cells, but the diffusion of the macrophages is becoming as one of the most sensitive parameters. The most sensitive parameters here in the case of the changes in the velocity. And then, so for the modeling of the treatment, we went back to the ODE model, and then we added the for FOLFIRI treatment is one of the most common chemotherapy treatments for colon cancer. So we added the FOLFIRI drugs that are basically 5-FU, LV, and IR. So irinotecan and leucovorin. So we added these drugs based on their published studies on how they interact with the components in the tumor. So those bold terms that you can see, these are the drugs that have been added, the equations for the drug, and how we modified the equations for the other players based on these drugs that have been added to the model. And we looked at the parameters of the model. We looked at the dynamics of the players in the model. And we saw that if we have only 5-FU, it doesn't work very well as the case that we have all other two other drugs as well. So if you look at the dynamic of the cancer, you can see that the cancer decreases at some point and then comes back very rapidly after the drug dosage has finished. But for the if you have FOLFIRI treatment for some of the clusters, that's basically here is cluster 3, we can see that it might take a long time to tumor get to this visible size. But for some other clusters, just rapidly again going up like cluster 2. We also looked at the treatments after 1 year or after 5 years to see if there is any difference between basically early detection and applying the early treatment versus the waiting for 5 years, after 5 years adding the treatments. And we saw that, interestingly, for some cluster does make a big difference, but not for others. So for example, in cluster 5, you can see that the tumor is slow growing. Even after 5 years, we have the added tumors. And then for cluster 4, it's growing very fast. Even it was after 1 year, it goes very fast. But for other treatments, just very fastly, the tumor would come back. And then we looked at how well the model would predict the accuracy of the treatment. So one thing is very important to mention here. We didn't fit this data at all. We fitted to basically the ODE model by assuming the large tumors are at the steady state. We didn't fit to the treatment at all. So the parameters of the treatments coming from the studies, observed studies, but the rest of the parameters is based on basically how the tumor grows over the time. But what was interesting, we saw that it was good, but not very good predictions of the tumors if they have been reoccurred or not in the first time of the follow-up, but it's very bad in the last time of the follow-up. The reason is that the tumor that has been reoccurred before is very different immune composition, and we didn't model that reoccurred tumor. We had modeled the original tumor at the beginning. So if you see that this is really bad prediction, it's because the original tumor was modeled, not the reoccurred tumor. And that shows that the reoccurred tumor might have very different immune composition than the original tumor. But this all still is not very good and excellent. So that's the reason we need to improve our models. So there are many other factors in the patients. It's not only the tumor environment that basically give us the response to the treatment. There are other stuff in the human body going on. So those stuff are also very important to be able to model and look at them. So for the next step, we would like to look at the MRI images, and based on the MRI images, get the boundary of the tumor, and then modeling the actual tumors, not basically the circle. And then also we are collaborating for the breast cancer with UMass to get the most sensitive parameters that we found, what are the most sensitive parameters for breast cancer, and also with Kate at Kentucky University for colon cancer to basically find the values of the most sensitive parameters for colon cancer and improved our model for these two cancer types. Also going back to the digital twins, I was also part of another team that got both funding from NCI and Department of Energy. And this model, it's basically it's more machine learning model. It's based on the graphical models and looking at the patients who have done a treatment similar to this and look at what is the closest patients, and based on the closest patient, finding the best optimal treatment option for the patient. One of the challenges of these types of the modeling, again, is lack of the data, because we might not have the exact same patients, very similar patients to in the observed data. But I believe that combining these two different approaches, like sort of ensembling them, could help us a lot. So we can have machine learning prediction in one side and also the mechanistic models at the other side, and then combine and having some weighted average would help us to basically overcome some of the challenges of these two types of modeling. So as I mentioned, the main challenges, lack of time course patient data. That's also right now, there are some potential simulations that there are more patient data are becoming available, like All of Us project and so on. And then also importantly, collaboration could help us a lot, because there are some other data that are in-house for some medical schools that we could help, we could use them. And then the other thing is several treatments that have not been, patients might have some treatments that have not been recorded. And this is another challenge. And then also it's hard to model all treatment and drugs. Even if we have their informations, then it's very hard to add. So to overcome of this challenge, we might be able to integrate drug pathways databases to the QSP model to help to overcome some of these challenges. And then for the data challenge, we can also come up with more generative models to create artificial data points. And by that, I would like to acknowledge both NCI and DOE and the public data sources. So thank you so much for listening, and any questions?